Science in Pajamas continues. Alright, so last time we were talking about how DNA is replicated in your cells in order to get an extra copy of it to put in cells that are going to be made during the cell cycle. So let's talk about the cell cycle. So a cell cycle is exactly what it sounds like. It is a cycle that the cell goes through. I know, shocking, right? And how that works is your cell has a job, whether it's a brain cell, a skin cell, or stomach cell, nerve cell, they have jobs. And most of the time, they're just doing their jobs, but every so often, you need to make more of a certain kind of cell. And that's where the cell cycle comes in. Now, the cell cycle is split up into two main parts. First, we have interphase. So we have interphase. And we have M phase. My kids know my handwriting sucks, so I apologize. So interphase is when the cell is doing its normal, everyday stuff. So that's not when it's actually dividing. Interphase consists of three portions. We have G1, S, and G2. Now, G1 is what the cell is, the stage that the cell is in after it finishes its previous cell division. So it's a brand new cell, it enters G1. There's actually a checkpoint at this point, or during this stage, to make sure that everything went well from the previous division. And during this time is when the cell is going to be making sure it's a functional cell. It's going to be making extra copies of any organelles if it still needs some in order to do its job. It's going to be taking in materials to grow. And it's also going to be just making proteins. Whatever kind of protein it has to make to do its job, that's the kind of protein it's going to make. So it's just going to be growing and doing its normal stuff. It's whatever job it's supposed to be doing. When it gets a signal that it is time to divide, or not that it's time, but we should probably start prepping, that's when it switches to S phase. S is for synthesis, and this is where the DNA is going to be replicated. So S phase is when the cell organizes its particular enzymes in order to make an extra copy of DNA. Now, once it's done with that, that's when we get to G2 phase. And this is essentially prepping for division, prepping for mitosis. So the cell is going to take in some added material. It's going to take in extra, whatever it needs to build, like the spindle fibers, some extra glucose in order to make ATP. And it's going to grow to be a certain size. So you want a cell that's large enough that when it splits, that the two daughter cells are still of a working size. So it's going to be growing some more and prepping for mitosis. Now, mitosis, as most of my kids know, because again, high school, you guys know that mitosis is cell division. And that's true. But what actually occurs after interphase, once we complete G2, we enter what's called M phase. Now, M phase, yes, the M stands for mitosis, but it's more than that. We have mitosis, which is the cell division, but then we also have something called cytokinesis. Now, cytokinesis will occur at the same time as the last part of mitosis, but it is actually not part of mitosis. Mitosis is talking about how the nucleus and the chromosomes are divided. Cytokinesis, like the prefix cyto implies, don't mind him, he's just barking at a neighbor dog. But as cyto implies, it is the division of the rest of the cell. So the cytoplasm, the organelles, and the pinching in of the membrane. Now, we're going to focus on showing the parts of mitosis. So let me erase my board. Mitosis has 4 steps: prophase, metaphase, anaphase, and telophase. So I'm going to start with the cell that is currently in interphase. Yes, not part of mitosis, but that's where we're going to start off as. Now, how this kind of works is I'm only going to draw the basics of what we need. Those little things right there are the centrioles. They're a particular organelle that really doesn't function except during division. During interphase, they're just chilling. And then we have our nucleus, and we have some cute little chromosomes. I'm going to give us 4 chromosomes, 2 pairs, a big and a small. Now, during interphase, your DNA actually is not in chromosome form. It's in chromatin, and that's when all the DNA is kind of super hyper condensed down. Think of it like a ball of string. If I take the string and stretch it out, it's super long, but super thin. And when I wind all the string up, it sits in a much more compact form. That's what the chromatin is. But I'm going to show the chromosomes so that you understand what we're starting with versus what we end with. All right, so now we're going to enter mitosis. First stage being prophase. Now, during prophase, we see a few things going on. We have our centrioles are starting to move and separate. At the same time, they're also building these things called spindle fibers. The nuclear membrane is actually going to break apart, which sounds weird because, we always talk about how the nuclear membrane protects the nucleus, and that's where we protect the DNA. But think about it. If you want to be able to move and separate the copies of DNA, then you need to take it out of that protective bubble. So that's what we're going to do. We have our DNA all separated into chromosome 4. A lot of times you'll hear in science books, it'll say the chromosomes become visible. Doesn't mean they were invisible before. What it means is the chromatin is so condensed down that it can't be seen with a regular light microscope, but chromosomes can be. So that's all that we need. Now, I know it's kind of hard to see, but how I draw the chromosomes are like this. That is one chromosome. Light won't mess that one up as much. So that's one chromosome. It is made up of 2 chromatids, sister chromatids. And what that means is we begin with 1, and then during S phase, you replicate it, and now you have 2 sets of the same information. Now, as we've discussed previously, and at some point I'll make a video talk about this as well. But my kids, we've already talked about this, which is why it's not in this video. Your chromosomes come in pairs because you get half of your genetic information from your mother and half from your father. So you have 23 pairs of chromosomes. That means that when we talk about homologous pairs, that's the same chromosome. It has the same genes on it. You just get one from mom and one from dad. Doesn't mean they're identical though. You might have different variations of those genes. So there could be a gene for eye color, but there's different variations. You can get a brown version from one parent, a blue eye version from another. That's for another section or another video. So we can see the chromosomes with their sister chromatids, nuclear membrane disappearing, centrioles beginning to move, and the spindle fibers are getting to form. Next, we go to metaphase. So in metaphase, the centrioles are now on opposite ends of the cell. Oh, Ripley. And they have attached the spindle fibers to the centromeres of the chromosomes. And they have stretched and pushed and lined them up so that the chromosomes are now lined up in the middle of the cell. Again, it's something we've already talked about with my class, but in case you forgot, the centromere is right here. It's right there. It is what connects the two sister chromatids to each other. So the spindle fibers are attached to the centromeres and they line up the chromosomes in the middle of the cell in between where the centrioles are located. Good way to remember that, metaphase middle. All right, then we go on to anaphase. And what happens in anaphase is the centrioles pull on the spindle fibers, and the spindle fibers will then pull on the centromeres of the sister chromatids, causing them to separate and split. So we have split the sister chromatids and we are now dragging them across the cell to opposite sides, towards where the centrioles are pulling them towards. So we're pulling to opposite ends. That takes us to the last part of mitosis, which is telophase or telophase. Now in telophase, which I have here, we now have two groups of chromosomes. And we are going to rebuild the nuclear membrane around them. As we rebuild the nuclear membrane, the chromosomes are actually going to go back into that chromatin format. The spindle fibers get broken down and recycled, or trashed, and that's the end of telophase. However, at the same time that telophase is occurring, this is also where we see cytokinesis going on. Again, cytokinesis is not part of mitosis, but it occurs at the same time as the last part, telophase of mitosis. And what that means is that the rest of the cell is going to start dividing. So the cell membrane is actually going to start pinching in. Pew, 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 pew. And it's going to keep pinching in and pinching in and pinching in. And while it's doing that, it's actually streaming and separating the cytoplasm to both ends, along with some of the other organelles that still have to be separated to the two soon-to-be daughter cells. So it's going to be pinching in until it meets in the middle. At that point, the membrane will fuse and the cell will pop apart. And we are left with two new adorable little daughter cells, each with the same exact DNA as each other, thanks to DNA replication, and the same DNA as the original parent cell from interphase. So now that we have two identical daughter cells, they now enter G1 of the cell cycle. So they are now in interphase. They're back where we start at G1. They're going to start growing, getting any materials they need to finish organizing and creating any additional organelles, checking to make sure nothing went wrong during division, and then making proteins to begin doing their cellular job. And that's essentially what we need to know about the cell cycle. So hopefully you found this entertaining, informative, and I guess that's 2 videos for today, so I'm probably going to do some other work on my computer. Hope this helps, and until next time, take care, everyone. All right, bye-bye.